Hello everyone. As you can see, uh, we will discuss about cell cycle. So, what is cell cycle? We will discuss cell cycle. Cell. What is first? We have to know that what is cell. Cell. It is like a complete life cycle of a particular cell, which we know as a cell cycle. Particular cell means one cell is completing their entire process, that is called cycle. So it's a uh, cycle of cell of a particular cell. So cell cycle distributed in a two stage. First one is interphase, and the second one is division phase, which we known as amphase also. So here we can see that interphase. In interphase, what we can see? In interphase, first is middle stage of cell cycle. Middle, because this, yeah, here you can see that middle stage of cell cycle, because on interface occurs between two division phase. So it is a middle stage of cell cycle. This is the middle stage, and it is the most active phase of cell cycle also. And in interface, metabolism of cell increases, and this stage was uh, occurs before mitosis, when mitosis begins. So at that time, this stage was occurs, and in cell, it grows the size and replication, and its DNA to repair for cell division also. So now we can uh, move toward the next slide. So here we can see that these two scientists, Howard and Pell. So just note it down. Just you can highlight this. So this is Howard and Pell. So they both are classified this interface, because interface has uh, interface grouped into three substage. So they both are classified into three substage. And what are those stage? So the stage are G1 phase, GS phase, and G2 phase. So as we know that G1 phase is occurs in synthesis of DNA. Synthesis of DNA means it occurs before S phase. So for that it's called pre-DNA synthesis. Okay, and it is also, we can say, gap phase also. So what we can see here in this phase, the most important thing is RNA and protein. Okay, this RNA and protein is the most important thing. So if you want, then you can note it down, RNA and protein. So whatever I have mentioned here, you can note it in your notes or whatever you will prefer. So in this phase, cells are synthesize RNA and protein both. And due to this protein, only particular protein, only particular this protein. So this is protein, okay. So due to this particular protein, what is happening due to this particular proteins, it is synthesized in cell division. In cell division of G1 phase, it is synthesizing with protein synthesizing, it is synthesizing and it creating the new protoplasm cell. Here it is creating the new protoplasm cell. And after then cell grows in a maximum form in G1 phase. So we can note this cells are So the number of cells are growing in G1 phase. And next one is S phase. So this is S phase. So S phase is also known as DNA synthesis phase. And in cell, here you can see in S phase, the cells are involved in several work like replication of a nuclear DNA and synthesis of histone protein. So here you can see replication. It is replicating the nuclear and synthesizing the histone protein. So this is important. You can make sure you have to note this in your notes that histone protein occurs in S phase and it is synthesizing because mainly DNA is synthesizing in this S phase and G1 and G2 phase are post and pre phase. You can easily you can remember that G1 phase is occurs before S phase and G2 phase occurs after S phase. Okay. So next what we have to see here the cytoplasmic DNA replications are also occurs in any stage of cell, but mostly in S phase. And the cell cycle can remain in G phase and G0 phase. Okay. So next one is uh, in G phase, we can see that the G0 phase found in cell of permanent tissue. G0 uh, found in the cell of permanent tissue. Okay. And the cell cycle of meristem cell is running by a special protein. So what is this special protein name? The special protein name is cyclin protein and CDKS protein. Okay. So here you can see and note it this cyclin and CDK protein. So these two proteins are running the meristem cell in cell cycle. Okay. Cyclin what and these two protein which are the prominent protein is cyclin protein. Why? Because cyclin proteins are target the DNA replication. Get my point? Because cyclic is that protein which is targeting the DNA replication and after DNA replication what will happen after DNA replication? After DNA replication it enter it into G2 phase. So cyclin protein is triggered that DNA replication. Okay. So now we have to see the DNA replication. So what it is? So you can, if you want, then you can note it down. And in G2 phase, it is also, as I said, that it is also post DNA synthesis and gap 2 phase. Okay. And why it is gap 2 phase? Because it is occurring after phase. So G2 phase actual cell divisions are occurs in this phase. This is very important. You have to remember that the G2 phase is actually occurs or actually division of cell is occurs in this phase only. And in G2 phase, what are the materials are required for the division of cell? Materials means some proteins are required, which is called tubulin protein. So you have to note it this tubulin protein also. Tubulin protein. This protein can help the spindle formation. You have to remember this. Tubulin protein is helpful for spindle formation also. This tubulin protein can help for spindle formation also. And what will happen after then? What happened after then? Cell divisions involves in anomalous expenditure of energy. So during G2 phase, energies are stored in the form of ATP. So here you can see that energies are stored in the form of so if you want to note it down. And 30 ATPs are required. 30 ATPs are required for one chromosome movement. Then it enters into amphios. After that, it enters into amphios. So in this phase, most cell division is most expensive for energy utilization. So it is most expensive. So for energy utilization, here I can mention this expensive expensive energy utilization. So uh, this is the important line, you can note it. And after the G2 phase, G2 phase, the cell enter into the division of M phase. So it occurs in after G2 phase. It occurs after G2 phase. So this phase, the cells are entered into M phase. Okay. So get this point. Now we can move towards next slide. Next slide is division of M phase. Division of M phase means short time in cell cycle, shortest time in cell cycle. This division of M phase, it is the shortest cell division phase or M phase. And in this phase, karyokinesis occurs, and karyokinesis is proposed by Schilder, and cytokinesis proposed by Whiteman. So just note it down, this one. Schilder. Okay. This Whiteman. It's important to remember because in many exams the scientist names are asked. And another, now we have to talk about now we have to learn mitosis. What is mitosis? First is that what is mitosis? What we can understand about mitosis is, so mitosis uh, refers to the spindle using nuclear division. A spindle using nuclear division, and it produces two identical daughter nuclei from the parent nucleus. If you want, we mention. If you want, you can note it here. So what about definition of mitosis? First, first to the spindle using nuclear division. Spindle using division. Okay. Use identical identical daughter nuclei. Um, daughter nuclei. from the parent. Okay. So yes, and it is producing like it refer to the spindle using nuclear division, and it produced two identical daughter nuclei from the parent nucleus. And another point is somatic cell division also occurs in this phase. You should know this. And we have that term Fleming was gave this term in 1882. Okay, and it is homotype and equational division. It is homotype and equational division. Segregation and recombination do not take place in this phase. Okay, segregations and recombinations are not takes place in this phase. So here we can see that coiling of chromosome. So here we can see that coiling of chromosome. Coiling of chromosome are two types, which is plectonomic and paranomic. Okay. So this chromosome, uh, what is plectonomic coiling? First, we can understand this. What is plectonomic coiling? So plectonomic coiling means two sister chromatids of a chromosome. Okay. Two sister chromatid chromosome are aligned together, aligned, which are similar to twisting of two strands, which are similar of two strands. Strand of the rope. To a strand of the rope. Okay. So it is called platinum coil. This is platinum. Two sister chromatids of a chromosome are aligned together, which are similar to twisting of two strands of the rope. Okay. And another one is paranomic coiling. Paranomic coiling is just erase this. And now paranomic coil. Paranomic coiling. So in this coil of one sister chromatid is simply slipped over the other sister chromatid, but not twisted to each other. Coils of, coils of sister chromatid, sister chromatid is simply slipped over the other sister, but twisted each other. Twisted each other. Okay. This is about paranomic coiling of this, and this both coiling are occurs in mitosis phase. Okay, if you want, then you can pause it and you can note it down. What we will study next. Next is sister chromatids are held together by multiple subunit protein with protein complex, which is called cohesion. This is important. You should note this one. So, so whatever I'm highlighting, you can note points. And the mitosis consists of four phases, as you know that. But yes, metaphase and telophase. So here, what we can see in prophase, it is the longest phase of mitotic division. Okay, the longest phase of mitotic division, and chromosomes are thin and long coil chromosome, which is called chromonimata. And the nucleolus and nuclear membrane are disappear at the end of prophase. So these two are very important one. Yes, as you can see in diagram also, nucleolus are disappearing. Nucleolus are disappearing. Okay, at end of prophase. This diagram is what this diagram is chromosome. Nucleolus at the end of prophase. The end of prophase, nucleolus are disappearing. Okay, here you can see. And the next is metaphase. So in metaphase, what we can see in metaphase, spindle tubes are formed and chromosomes are oriented at equatorial plane. So this is very important. Equatorial plate. Spindle tubes are formed and the chromosomes are oriented and oriented where? Oriented at equatorial plate. Okay. Sorry, just to wait. Okay. Sorry guys for the interruption. So what we will in the next. Once it attached to the spindle fiber, this cohesion is degraded. So in the previous slide, what we have studied about cohesion, cohesion means protein. Okay. So degraded by enzyme, which is called separase. Okay. What is degrading in this stage? The spindle fiber. Once a spindle fiber to attach, once it attached to a spindle fiber, cohesion is degraded by enzyme. This cohesion, this protein is degraded by another enzyme which is called separase. Okay. So next is then sister chromatid are disjoint except the centromeric region due to protein called sugar shell. Okay. Now this also illusion. So sister chromatids are disjoining, are disaligned, and accept the centromeric region. Accept at centromeric region. Accept this centromeric region. Uh, why it is accepting the centromeric region? Sister chromatids are disjoining. Okay. Sister chromatids. Disjoining. Sister chromatids are disjoining. But accept this centromeric region. It is not disjoining or disaligning the chromosome. Why? Because of this Shugoshin. Shugoshin is Shugoshin, Shugoshin because of this Shugoshin. And what is Shugoshin? You have to know about this Shugoshin also. Shugoshin also a kind of protein. Okay, a unique protein. It belongs from a unique protein family, and it protects the cohesion from degraded by separase at the stage of centromeric region. Okay, I think you understand. It protects the cohesion. Sugosine is protecting the cohesion, protecting the enzyme or protein, okay, protecting the cohesion from being degraded by separase. Because separase also enzyme. Separase here you can see that separase is enzyme. So cohesion is protected. This protein is protecting to degraded that centromeric region. Centromeric region is not degrading, and the centromeric region is not separating. Why? Why it is not separate? Because centromeric region here you can see centromeric region is not separate. See this centromeric region in this diagram. Okay. This is centromeric. Centromeric region is not separating because of what? Shugoshin protein. Because of Shugoshin protein. Okay. So this protein is protecting how it's work. You know that protecting protecting the cohesion, protecting the cohesion from degrading, protecting cohesion to degrade. Yeah. So what it is protecting that cohesion. It protect the cohesion to degrade by separase. Cohesion is protected. And how it is protecting? It is protecting through this Shugoshin protein. This cohesion is working as an adhesive. Adhesive means we can use something for joint. So it is work as an adhesive. Okay. So note this. This adhesive, this cohesion is protected by Shugoshin protein. So it's a double protection as it is. Double protection. So you can easily note this. And after Shugoshin, chromosome are biggest and smallest and very clear. As you can see, it is very clear. Chromosomes are very clear. and it is the best stage to study the chromosome. Why? Because of this clearness, because we can see, or it is visible very clear. It is clearly visible. Okay. And next is anaphase. So anaphase is, what is anaphase? In anaphase, we can say a centromeric region of homologous chromosome divides, and it result as a chromatid, and it result as a chromatid separate at centromere and moves toward the opposite pole. Okay. Here you can see this stage ensure that each cell contain identical chromosome after splitting, and chromatid separate and moves towards the opposite end of the cell. Okay. So here are the chromatids. These chromatids are moving toward the opposite pole. Okay. Towards opposite pole. And centromere of homologous chromosome divides and then chromatids separate at centromere. Chromatids separate at centromere. After separation, each chromatids become chromosome. Okay. So next is telophase. Telophase. What is telophase saying? Telophase in this cell division, two nuclei form around each set of chromosomes. Two nuclei form around each set of chromosomes. This is going to form set of chromosomes. Chromosomes decondense at the cell begins to split from each other, and it is also called a reverse. Because of this opposite direction, it is also called a reverse prophase. And the spindle tubes disintegrated, nucleus also reappear at its pole, and the chromosome are uncoiling and unfolding longer and thicker, and it's look like thread and light microscope. So we can see this chromosome. It's look like thread like a structure. Okay. And last is cytokinesis. What is cytokinesis? It is a division of cytoplasm, which is known as cytokinesis, and it takes place in two cells, in animal, in plant cells and in animal cells. And cytokinesis, here we can see that in animal cell and in plant cell, the cytoplasm of the parent cell is split from two identical daughter cells. This is two different daughter cells. Different identical cytokinesis. So after this cytokinesis, we have to uh, enter into meiosis. Okay. So in cytokinesis, I can explain this. In cytokinesis, in plant cell, cytokinesis works the formation of cell plate, and it lay down across the region of metaphase plate. Lay down across the region of metaphase plate. So it in plant condition, okay. And in animal, cytokinesis is through formation of cleavage. Curvature of cleavage furrow or constriction. In plant, cleavage furrow is not possible due to the presence of ridged cell wall. Okay, cleavage furrow is not possible, but in animal, cleavage furrow is possible. Cleavage, cleavage furrow. Furrow. But in animals, it in your mind, it's a step is meiosis. Okay, so study the meiosis later in the next part of cell cycle. Okay, so here you can see that whole process is also stored in the opposite pole, and in metaphase it is sugar sugar protein. Here you can see that the plate like a structure. In the light microscope you can see this telophase you can see very clearly under light microscope. Under light cytokinesis, it's in plant, in animal, in plant. What is the main important point in plant? In plant, cleavage flow is not possible. Flow is not possible. In animal, it's possible. So, can make this clear notes. Of cell meiosis. Discuss it later. So, prepare your notes. Okay. Thank you so much for watching. Bye bye. We can see you.